everyone. My name is Divya Sharma. I am a research scholar at Department of Biotechnology, Indian Institute of Technology, Madras, India. The title of my research is Elucidating Important Structural Features for the Binding Affinity of Spike SARS-CoV-2 Neutralizing Antibody Complexes. COVID-19, as we all know, has affected the lives of millions of people around the world with nearly 540 million confirmed cases and nearly 6 million deaths. The SARS-CoV-2, which is the causative agent of COVID-19, has spike protein which contains the receptor binding domain that binds to the host cell receptor ACE2 and mediates the entry into the host cell and cause infection. Researchers around the world have developed neutralizing antibodies that target the SARS-CoV-2 spike protein and block the binding to the host cell receptor ACE2. There have been mutations in SARS-CoV-2 spike which led to the emergence of new variants such as alpha, beta, gamma, delta, and omicron, and has also led to immune escape from the neutralizing antibody. Still, there is a need for large-scale analysis to understand the biophysical factors leading to decreased or loss of binding to the neutralizing antibodies. So we analyzed the interface of spike and neutralizing antibody complexes, and our work provides insights into spike antibody interactions, structural parameters related to the binding affinity, and mutational effects on binding affinity change, which can be helpful to develop better therapeutics against COVID-19. Here, figure 1 shows on the left SARS-CoV-2 receptor binding domain bound to host cell receptor ACE2, and on the right, SARS-CoV-2 receptor binding domain binds to the neutralizing antibody CD6 with the heavy chain and the light chain. Coming to the methodology, figure 2 shows the overview of the methodology. We obtained 29 SARS-CoV-2 spike antibody complexes from protein data bank and calculated structural features such as propensity, pair preference, interaction energy, interface area, surrounding hydrophobicity, etc. And we tried to develop a machine learning model. As a second part, we did the mutational analysis of the interface where we looked at the mutations that decreases the affinity and stability of the epitope and the mutations that increases the affinity and stability of the paratope and compared our results with the known variants. Coming to the binding energy, the experimental binding affinities were obtained from the literature, and the binding free energy delta G was calculated from Kd binding affinity using the equation delta G is equal to RT ln Kd, where R is the gas constant and T is the temperature. Coming to the calculated features, the binding propensity was calculated as the ratio of the percentage of residue I in interface to the percentage of all residues in the interface. Residue pair preference for the spike antibody residue pairs at the interface was computed using the following equation, where I and J stand for the interface residues in spike and antibodies respectively. Nij is the number of interacting residues of type I in spike and J in antibodies. Ni and Nj are the number of residues of type I in spike and J in antibodies respectively. The interaction energy, atomic interaction free energy between the interface residues of the spike and antibodies was calculated using Ember potential, and the equation is shown with the following equation. Accessible surface areas of the RBD region of the spike protein SE and the antibody is H plus L, as well as the entire complex, which is S complex, were calculated by rolling a water molecule of radius 1.4 angstrom over the protein or the complex surface. And further, the interface area was calculated as the subtraction of the surface areas of the complex and the free proteins. The surrounding hydrophobicity of the ith residue of the protein Nij was calculated using the following equation, where Nij is the total number of surrounding residues of type J around residue I within 8 angstrom distance between C alpha atoms. Hj is the hydrophobicity value for the residue type J in kcal per mole, which is obtained from thermodynamic transfer experiments. For mutational analysis, we calculated the changes in binding affinity and stability using servers such as MCSM, Foldex, CupSet, and ProFemusic, and we combined their results to obtain the mutations that decreases or increases the binding affinity. Coming to the results, Figure 3 here shows the propensity of interface residues for spike and antibody. From this figure, we can see that tyrosine had the highest propensity to be at the interface. Figure 4 shows the energetically favorable pairs which are most preferred at the interface. We found that tyrosine containing contacts were most preferred at the interface, and from these results we concluded that the aromatic interactions are important for binding of the spike protein to the antibodies, to the neutralizing antibodies. We also developed the regression model. Uh, Figure 5 shows a regression plot showing the relationship between experimental and predicting binding energies on training and jackknife test. And for training, we obtained a correlation of 0.93 and mean absolute error of 0.48 kcal per mole. And for jackknife, we obtained a correlation of 0.9 and mean absolute error of 0.53 kcal per mole. Figure 6 shows the examples of the mutations of two mutations, E484K and K417L, K417N at the spike interface. From this figure, we can see that upon mutations, the interactions between the spike and the antibodies are lost, and the binding affinity is also decreased by 1.83 kcal per mole and 1.23 kcal per mole as shown by the experiments. Coming to the conclusion, our results conclude that propensity of tyrosine was observed to be the highest at the interface of spike antibody complexes. The structural features that we identified could predict the binding affinity of SARS-CoV-2 spike to neutralizing antibodies with a correlation of 0.93. And the mutational analysis revealed the sites that could increase or decrease the binding affinity of spike protein to the neutralizing antibodies. Further, I want to acknowledge Indian Institute of Technology Madras and RBCDSI for the grant. And our work was published in Protein during early 2020. Thank you.